All right, so let's get started. Hello, everyone. Welcome to our today's Zoom seminar. It's absolutely my pleasure to welcome our today's speaker, Dr. Taras Slavsky. Taras is mainly interested in study T cell biology and T cell development, and a very, very brief introduction about Taras. Taras completed his undergraduate study in Bachelor of Science in Biology, and then continue with his master's degree training in immunology, both back in Russia. And then he decided to pursue his scientific career and then get the training in immunology program as a PhD student in Harvard Medical School in Boston. And then after that, he completed his PhD training in immunology and then moved to Europe from 2013 as a postdoc fellow, get the training in Research Institute of Molecular Pathology in Vienna, and then did a second postdoc back in Boston, also in Dana-Farber Cancer Research Institute. So from 2018, he actually moved to Sweden, started his independent research program, as we probably can see from his publication. He moved to Karolinska Institute in Stockholm in Department of Medicine, Solna, and then start his research program as a laboratory of lymphocytes biology. So Taras Lab is mainly interested in studying the machinery of cell fate decision made by innate-like and then conventional lymphocytes. And then the study direction in his lab is including two different directions. One, his lab is studying mechanisms of cell fate decision in humoral immune responses underlying the formation of immunological memory. And then the second part is how the specificity, development, and the function of innate-like lymphocytes. I believe today we're going to hear Taras' recent work regarding how the specificity and function of innate-like T cells in the thymus. So welcome, Taras. We are very much looking forward to your talk.
Many thanks. Many thanks for inviting me, and many thanks for this very kind introduction. So today I will tell you two stories focusing on our T cell projects, both about innate-like T cells. One about very strange functions of the cells, and the second one, unpublished story about very strange antigen receptor specificities. And the first story was driven by these two talented people, a former postdoc in the lab, Josephine Dunst, and in the first place, a former PhD student, Yuan Yu. And this story started away from from innate-like T cells. We asked ourselves a question about formation of tolerance to a rather unusual class of self-antigens, namely the molecules that are normally induced upon inflammation. Because if you look at the sort of very schematic view of how T cell tolerance works, in the thymus, if developing thymocyte sees the antigen presented by thymic dendritic cell or by thymic epithelial cell, it is eliminated either physically or functionally. Then if naive T cell encounters an antigen in the context without inflammation at the periphery, it is also supposed to be eliminated, presumably by function, at least functionally, so it doesn't cause autoimmunity. And only in the context of inflammation that is usually triggered by presence of pathogen, you have you give a license to a T cell to respond. And this response, of course, involves multiple molecules that are upregulated both in innate immune cells and also in adaptive immune cells, including cytokines, cytotoxic molecules, costimulatory molecules, and so on and so forth. And these molecules coincide in time and space with molecules of the pathogen. And this, of course, should represent a unique challenge for the adaptive immune system to discriminate between the two. And when we started this project, nothing was known about the mechanisms and time point at which these cells that would be recognizing something like interferon gamma or IL-4 or granzyme B would be eliminated. And we thought it's an interesting question, and we decided to focus on that, mostly focusing on the molecules that come from activated T cells themselves. And if you look at the expression pattern of these molecules in the thymus, and this is just ImGen database, and you can appreciate that these are all different thymic subsets, and you can appreciate that thymic medullary epithelial cells express some levels of these inflammatory mediators, but the highest levels of these molecules are expressed by thymic innate-like T cells, including iNKT cells and some gamma delta T cell subsets. And what was also interesting about the cells, that some recent papers from Kreslavskiy's group and from Ima Prince's lab indicate that many of these innate-like effectors in the thymus are not newly developed cells. These are the cells that are thymus resident, that developed months ago and stay there more or less eternally. And the reason for accumulation of these effector cells in the thymus, at the place where you would think that initial pathogen encounter seems rather unlikely, remains somewhat enigmatic. And we started to think, what if the cells are present there, at least in part, to induce tolerance to this type of effector molecules that they express? And to start addressing this question, we focused on the mice that express model inflammation self-antigen, so to speak. We used both IL-4 GFP and IL-17 GFP mice with very similar results. This is published, so I will be focusing for today's talk on only on IL-4 GFP. And you can appreciate that if you look at the thymic innate-like T cells, more than half of the iNKT cells, for example, express the IL-4 GFP reporter. So we decided to see whether these molecules in general first induce any T cell tolerance. And to address that, we first just immunized the mice with an MHC1 epitope from GFP. And if you do it in wild-type animals, you see very nice expansion of GFP-specific CD8 T cells. But if you do that in alpha GFP mice, this is completely abrogated. They look like unimmunized animals, indicating that there is very strong tolerance to this epitope in the system. And of course, in this case, GFP can be coming both from hematopoietic cells and also from stromal cells. MTECs express a little bit, medullary thymic epithelial cells express a little bit of alpha GFP. So to test if hematopoietic cells are sufficient, we also performed similar experiments, but in bone marrow chimeras, where we transferred IL-4 GFP bone marrow into wild-type animals, and the result is exactly the same. If you immunize these chimeras, there is no response, indicating that GFP, this IL-4 GFP expressed only by hematopoietic cells, is sufficient to induce very efficient tolerance. So now the question is whether this happens already in the thymus. And to address that, we took advantage of a very nice mouse model that was developed by Brian Brown's lab, JEDI, that's actually a TCR monoclonal mouse that recognizes exactly this CD8 epitope from GFP. And we can mix bone marrow chimeras with this mice. And if we mix JEDI bone marrow with wild-type bone marrow and transfer into wild-type recipients, so there is no GFP in the system, you can nicely find the cells back in the thymus using here a congeneric marker. You can see that they express the TCR that you expect them to express. They recognize this GFP epitope. And then if you look at their development, it looks they behave as you would expect MHC1 restricted thymocytes behave. They differentiate from double positive to CD8 single positive. But what happens if we instead mix JEDI bone marrow with IL-4 GFP and transfer again into wild-type recipients? So in this case, all GFP in the thymus would be only coming from hematopoietic cells. The picture changes drastically. We basically completely lose CD8 single positive cells recognizing GFP. And this happens already at double positive stage. The majority of double positive cells are wiped out. And we know, I don't show you this data here, but we know it's cell death. We know it's negative selection. So it looks like there is a very efficient elimination of this constant stream of TCR transgenic cells in the system. And this is so efficient that even at the periphery, you barely find any GFP-specific cells. And to give you full picture here, of course, I told you that GFP is also in alpha GFP system expressed a little bit by thymic stroma, and indeed expression by the stroma population only in the setting where we use alpha GFP recipients and wild type plus JEDI bone marrow. We see the same picture. So it's a very redundant system. We can very efficiently eliminate in both cases all GFP specific cells, suggesting that maybe elimination of such inflammation-associated self-antigen specific T cells is very important in the immune system. That would be one interpretation. But if we look at the hematopoietic compartment, what are the cells that mediate this elimination of the constant stream of TCR transgenic cells? Turns out that it's a very small population of cells, something between 0.1 and 0.5% of total thymic cells. And as we expected, it's mostly innate-like T cells. But in addition to innate-like T cells, there was a sizable fraction of thymic eosinophils that expressed GFP. And because eosinophils, although they're not famous antigen-presenting cells, are myeloid cells, you start to think, okay, maybe what we're seeing is just mediated by eosinophils, and innate-like T cells do not contribute to tolerance. To test if this is the case, or innate-like T cells are sufficient to induce tolerance, we did multiple experiments, including thymic cultures, reaggregated thymic organ cultures, RTOX, which is basically an organoid that you can form by taking fetal thymi, digesting them, and then you can spike in any populations you're interested in. And we spiked in Jedi double positive thymocytes, sorted, and then sorted iNKT cells either from wild-type mice or from IL-4 GFP mice. And then you can assemble these organoids and keep them for a week or so in culture and see what will be happening there. And turns out that if you have wild-type iNKT cells plus Jedi thymocytes, you can nicely find Jedi cells back. And what I'm not showing you here, they progress during these days from double positive stage to CD8 single positive stage, as you would expect. But what happens if you spike in IL-4 GFP expressing NKT cells? So here, the only GFP would be coming is GFP derived from iNKT cells. And we spiked them in reasonably physiological numbers that you see in the thymus. Turns out that this is sufficient to nearly completely eliminate Jedi cells, and few remaining Jedi cells upregulate PD-1, indicating that they actually saw the antigen. And again, I don't show you this data, it's in the paper, it's a highly redundant system, so if you do the same experiment with eosinophils, they also work. So it looks like it doesn't actually matter for CD8 T cell tolerance much what is the cell that expresses the antigen, many different cell types can do it. And then the question that we got from the reviewers was, okay, this IL-4 GFP in the system is IRIS GFP, so it's intracellular protein. What happens to the real secreted protein? And to address that, we cloned constructs, retroviral constructs with either secreted or intracellular GFP, infected iNKT cells with these constructs, and then checked them in the same system. And both secreted and intracellular GFP can eliminate the autoreactive cells. But of course, you can also say that most likely this Jedi TCR has very high affinity to this GFP epitope. So could it be that this is an idiosyncratic feature of a setting where you have very high affinity TCR? To test this, we switched the system a little bit. We still use RTOX, but instead of spiking in double positive thymocytes from Jedi mice, we took OT1 double positive cells. And OT1s recognize an epitope in ovalbumin, and ovalbumin, the people describe mutations in the epitope that change the affinity of the interaction with OT1 TCR. And we can spike in then wild-type iNKT cells transduced with the retroviral constructs expressing different ovalbumin variants into the RTOX and see what happens there. And if you start with high affinity epitope version, the system behaves exactly as we saw for Jedi. OT1 cells are eliminated. But what happens if we go down with affinity? Turns out that you can go down up to 700-fold with affinity, and you still see negative selection. This ultra-low affinity version of OT1 epitope, you have about half of the cells eliminated, but if you go 70-fold down, you basically have eliminated almost all OT1 cells, indicating that this mechanism functions across very broad range of affinities. But this is, of course, in vitro. What about in vivo? So here we can, again, go back to our IL-4 GFP system, sort iNKT cells from GFP, from IL-4 GFP mice, and then transfer them intrathymically. So inject them directly into the thymus. And in this case, we inject them intrathymically into mice that have Jedi TCR beta chain without TCR alpha. And in this setting, without any transfer, you see a small but stable population of GFP-specific cells, because alpha is polyclonal here. Only a small fraction of alphas would result in GFP-reactive TCRs, but this is way above what you see in a wild-type setting. So this is a more physiological setting to address it than full-blown Jedi TCR, but at the same time, it's sizable population, so you can monitor what happens to the cells. And when we transfer L4-GFP iNKT cells, the first thing that we check 7 days later is whether they're still there. And yes, they engraft. And this was nicely, we were following the protocol that was designed by Chris Hockvist's lab. And indeed, the cells can be found back. But the numbers that we can achieve in this system is about 5 to 10 times lower than what we see in steady state. So it's really homeopathic amounts of the cells. And turns out that this very small numbers of cells is sufficient to eliminate many of these Jedi-specific cells, and the remaining cells upregulate PD-1, indicating that they saw the antigen. So it's a remarkably efficient system. And at this point, I started to think that most likely, because we thought the cells stressed, they should be dying, both for RTOX and here, that most likely we see some effects of cross-presentation, that professional antigen-presenting cells just pick up GFP, and that explains the efficiency of the negative selection. And we did multiple experiments there in the paper. I will be just showing you one example. We did MHC mismatched RTOX, bone marrow chimeras. In all the settings, it looks like it's direct T to T cell antigen presentation. And I will show you the most visual way to evidence for that. And this is imaging of RTOX that we performed just to be able to see this contact antigen presentation events between NKT cell and Jedi thymocytes. So we sorted L4-GFP NKT cells that will be in magenta here, and then sorted Jedi double positive cells, put them in RTOX, but before we loaded Jedi double positive thymocytes with calcium sensor dye. So whenever TCR signaling happens, we see firing of this calcium sensor. And now let's see if the video plays. Yeah, it does. So you can see there is a contact, and then the cells start firing, right? And then there is even some chase of Jedi cell follows iNKT cells and keeps receiving this TCR signal. So it was quite amazing to see. And then they show up again, and they continue to interact. And this, of course, only happens if you have GFP in the system. If you have wild-type NKT cells, nothing is happening. So this tolerance, at least in part in our system, it seems like in all the cases we tested, it seems to be happening by some reason through direct T to T cell antigen presentation, which was quite unexpected. But of course, that's all very interesting, but both ourselves and our reviewers were concerned that this is all shown with model antigens. And we decided to try to find evidence if similar mechanism functions to a real rather than model inflammation-associated self-antigen. And to do that, we decided to focus on interferon gamma, which in the thymus can be expressed a little bit by TAX, as a lot of other things. But most of it, as I showed you in the very first slides, and now I'm showing you here, is expressed by thymic effector lymphocytes. By some of them are T cells, NKT cells, probably myeloid cells, gamma deltas, and so on. And some of them are ILCs and NK cells. And so what we did was screened interferon gamma for predicted class 1 epitopes. And then we found an epitope that causes very nice response when we immunize interferon gamma knockout mice. But there is basically almost no response in wild-type animals. So these are mice that were immunized, and then we re-stimulate the cells in vitro with the peptide after immunization and use TNF-alpha as a readout for antigen specificity in this case. So this indicates that there is a tolerance formation to this epitope in wild-type mice, which is broken, of course, when you don't have interferon gamma anywhere. So now the question is, what happens if you have interferon gamma only coming from hematopoietic cells? And to this end, we used the same trick as I showed you before with L4-GFP. We used chimeras, wild-type into wild-type, mimicking wild-type setting, knockout into knockout setting. And then this group where you have wild-type bone marrow transferred into interferon gamma knockouts. So in this case, all interferon gamma will be coming only from hematopoietic cells, and that means mostly from ILCs and innate-like T cells. And if we immunize these mice, turns out that this expression by hematopoietic cells is sufficient to induce tolerance, basically completely abrogating the response. Of course, we don't know in this setting if it happens, and to which extent it happens in the thymus, but together with the results that I showed you at the previous slides, I think we're pretty confident that at least some of this tolerance occurs already during T cell development in the thymus. So what I showed you in this first part of my talk is that innate-like T cells are potentially ideal libraries of T cell-derived inflammation self-antigens because they are thymus resident, and we think they stay there for this reason, at least in part. And they produce molecules associated with Th1 inflammation, Th2 inflammation, Th17 cytotoxic responses, and so on. And moreover, very small numbers of the cells can induce very efficient CD8 T cell tolerance, which, at least in the settings that we checked, happen quite surprisingly to me. That was not something I was expecting through direct T cell to T cell antigen presentation. And we think that this mechanism is just one example of how thymus induces tolerance to inflammation-associated self-antigens. Because if you check literature or if you check expression datasets, for example, this is expression dataset unreleased yet from Spatial ImGen project, you can appreciate that almost everything related to all sort of inflammation pathways, including type Y cytokines, type 2 cytokines, interferon signaling genes, MyD88 signatures, S100 family members, and so on and so forth, almost everything is expressed in the thymus. And this is not just ectopic expression by MTEX. What happens is that this cells involved in these pathways form very complex networks in the thymus. This is just one example from a figure from my recent review, only focusing on type 2 inflammation. And the same would be true for other inflammatory pathways as well. And you can appreciate in the thymus, you have very complex interplay between thymic Tuft cells, iNKT cells, thymic B cells that differentiate to plasma cells, switch to IgE. There are ILCs there are specialized dendritic cell subsets, eosinophils, and so on and so forth. So we think that what we showed in this story that I just shared with you is just the tip of the iceberg. And in general, thymus evolved as some sort of low-grade inflammation site, if you will, or at least a site where you have spontaneous constitutive activation of virtually all inflammatory pathways, and that we think is turned on for educational purposes. Actually, Chris Hockfist had a very beautiful paper in Science Immunology about the role of type 1 interferons in formation of tolerance, that are one of the pathways that is activated in the thymus. And then a second speculation that I want to make here is that in addition to ectopic antigen expression by medullary thymic epithelial cells, I think we should also think about ectopic cell types present in the thymus. Because if you check the literature, in the thymus you have B cells, you have neutrophils, eosinophils, ILCs, NK cells, and many other small populations. And we think that one of the reasons they come there, or they stay there, is to induce tolerance to their own sort of slices of self. And many of them also undergo activation in the thymus and contribute to these inflammatory pathways. So now I switched gears a little bit from this published story to an unpublished story on very unusual antigen receptor specificities of innate-like T cells, and specifically gamma delta T cells. And this story was spearheaded by extremely talented PhD student in the lab, Kiwi Ye. Many other people also contributed to the story, including Josephine that I mentioned before, and Nimi Francis that also helps with the story right now. But many other people, of course, were also involved. And here, if you look at the adaptive immune system of all jad vertebrates, from cartilage fishes to mammals, you find three types of adaptive lymphocytes defined by their antigen receptors formed by VDJ recombination. You have B cells, alpha beta T cells, and gamma delta T cells. And for B cells and alpha beta T cells, we understand well the rules of antigen recognition, we understand the nature of the antigens, and because of that, we understand the function of the cells. For gamma delta T cells, this is not the case. We actually don't quite understand why the cells have TCRs in general. And in most of the cases, we don't know what these TCRs are capable of recognizing. What we know is that many of gamma delta T cells already in the thymus induce effector programs. And at least in some cases, we have good evidence that this effector program induction is driven by recognition of endogenous ligands for their TCRs, endogenous antigens, if you will, expressed in the thymus. In most of the cases, this existence of these ligands is pretty much hypothetical, but in a few cases we know the ligands, and they fall roughly into two different categories. Some rare gamma delta T cell subsets seem to be recognizing something MHC related. To me, it's not clear whether there is any focusing of a small fraction of gamma delta TCR repertoire on MHC-related molecules, or whether we expect a T cell to recognize something MHC-related. We have good tools, and we fish out few cells that actually do recognize MHC-related molecules. But at least this recognition usually involves CDR3 loops formed by VDJ recombination. Now, the second class of ligands that became very popular in the recent years through work of Adrian Haidey and many other labs are butyrophilin-related molecules. And these molecules function in a completely different way. They interact with germline encoded sequences of the gamma chain and more or less function as superantigens, or at least non-clonal ligands that allow to activate or induce functional maturation or particular tissue localization of large gamma delta T cell subsets that utilize particular V gamma chain. And the importance of gamma delta of the TCR composition for the gamma delta T cell biology can be illustrated by a fact that a lot of gamma delta T cell subsets, functional subsets, are defined by a particular combination of V gamma and V delta chains. And here are some examples from mice. There are also some examples in humans. But if you look at recent repertoire sequencing studies in mice, one of the most frequent chain pairings at the periphery at secondary lymphoid organs, but also in the lung and in the liver of mice, are V gamma 4, V delta 7 gamma delta T cells. And actually, even if you work on gamma delta T cells, you may never heard about these cells because there is no V delta 7 antibodies, and people till recently didn't pay much attention to the subset. And I come back to the cells in a few slides from now. But what we decided to do, because so little is known about gamma delta T cell reactivities, we decided to try to take a semi-systematic approach to the problem. We cloned a bunch of gamma delta TCRs from many different subsets, mostly focusing on innate-like effector subsets, which may undergo agonist selection. And then we plug them into reporter cells that have everything required for TCR signaling except for TCR itself. And these reporter cells have upon TCR signaling induced a fluorescent protein that first shines in blue, and then with time it gets oxidated and shifts the fluorescence to red. This second part is not so important. It allows to clean the background a little bit, but the most important thing here is that when you see a plot like this, that means that the cell just went through TCR signaling. And then we took these 55 cell lines that are identical except for the TCRs that they express, and co-cultured them with different cell types that you can find in thymic microenvironment. And when we did this exercise, and we keep doing that, but so far out of these 55 TCRs, we found at least 19 that were reactive to something. So for example, this TCR here, this is different TCRs, 7 examples here, and these are different cell types that we think may correspond to what developing T cell might see in the thymus. And you can see that this TCR, for example, fires in response to endothelial cell line and nothing else activates. These are just FACS plots from this is just flow cytometry after co-culture. And this TCR, for example, is slightly more promiscuous. And if you look at the patterns, you can appreciate that patterns between TCRs are quite different, indicating that there should be multiple different ligands responsible for this reactivity. And of course, the big question here are those previously known gamma delta TCR ligands, or at least ligands that belong to known families. And to address this question, we can take advantage of the fact that a lot of our TCRs converged in their reactivity on thymic double positive lymphoma cell line BICA cells. Many of them also recognized actual double positive thymocytes. And we decided to check what these BICA cells are expressing. And turns out by just doing bulk RNA sequencing, we can see that they don't express anything remotely butyrophilin related. They don't express or express very low levels of some other molecules that were implicated as gamma delta TCR ligands either in mice or in humans. But they, not surprisingly, express a bunch of MHC1 related molecules. So could it be that the reactivities that I showed you on the previous slide are explained by MHC1 related things? To address this, we can use a simple trick. We can just knock out beta-2 microglobulin from BICA cells and then repeat the co-culture experiments. And we did this exercise, I think, for 11 or so BICA reactive TCRs. I'm showing you 6 examples here. And from those 11, we saw only one that lost the reactivity to BICA cells if we remove beta-2 microglobulin. And here we rapidly could figure out that these are the previously identified non-classical MHC1 molecules, T10 and T22, that could activate this TCR. But the exciting result here is that that means that these diverse reactivities that I showed you on the previous slide cannot be explained at all by known classes of gamma delta TCR ligands. So there should be something new here. And we're trying to understand what can that be, what kind of molecules gamma delta T cells can be selected on. And here we were successful 2-3 times with an approach that is called cDNA library screening. And actually, This is the approach that we borrowed from after reading Alsinger's papers about where he was trying to understand the reactivities of very unusual alpha beta T cells that develop in the absence of MHC and co-receptors. And here the idea is very simple. You can take ligand expressing cells. For us, it was Beaker lymphoma cells that we focused on that activated many different TCRs. We can make mRNA, and then we can produce cDNA and clone it in bulk into expression vector. And then we can transform E. coli and titrate down them in such a way that we have about 200 clones per well of a 96-well mini-prep. And then we can just transfect the feeder cells with these pools and co-culture them with our reporter cells. And if the ligand can be encoded by single cDNA, which is of course a big limitation of the approach, we wouldn't be finding anything that would be heterodimer or would require some complex processing and so on. But if the ligand can be encoded by single cDNA, then we should be able to see the activation of the TCR signaling by just flow cytometry, and then we could see what is the molecule behind it. So that's one example. I think we found 2-3 ligands so far. I will be focusing only on one today. And here you can see that there is, this is just a 96-well plate. Each dot is one well, and you can see that the frequency in reporter cells was very high in one well and low in the others. So then we can, through serial dilutions, get to the single cDNAs from this pool. And we can just Sanger sequence what are these single cDNAs that are responsible for this reactivity. And when we did that, we found something that you wouldn't expect to be activating TCR signaling, because it's a cytokine receptor. It's IL-17 receptor A chain that was responsible for induction of an FAT reporter here. Of course, from this experiment, strictly speaking, you can't even say whether this NFAT reporter activation happened because of TCR signal. So to test this, we now compared the first TCR that we used in the screen, and this is a V-gamma-4 V-delta-7 TCR. You remember I mentioned this particular chain combination earlier, and it's nicely activated again by IL-17 receptor overexpression, but the same reporter cells expressing a bunch of other TCRs are not activated at all, which now formally demonstrates that what we see is a TCR-mediated, TCR-specific reactivity. Moreover, what was very exciting in this experiment was that Kiwi found a second TCR in our relatively small collection, which was also a V-delta-7 TCR, this time with V-gamma-1, that was also activated by IL-17 receptor, and that indicated that maybe that should be a reasonably frequent population in vivo, because we found 2 TCRs in relatively small subset that we analyzed. Then as a sanity check, of course, we knocked out IL-17 receptor from Beaker cells, and sure enough, the reactivity was gone. But even more excitingly, when we use just recombinant IL-17 receptor protein, this is sufficient to induce the reactivity of these TCRs, but not of the other TCRs. And that, of course, immediately gives us hope. So IL-17 receptor is not only required to activate this TCR signaling IL-17 receptor A chain, but also sufficient. And this, of course, gives us immediately hope that we will be able to use IL-17 receptor as a staining reagent to fish out the cells that are recognizing it in vivo. And it sounds very simple. It turns out to be much more complicated, because it's probably quite a low affinity interaction. So Kiwi had to find some ways to multimerize IL-17 receptor to very high degree, but he managed to generate IL-17 receptor multimers that stain nicely reporter cells that express IL-17 receptor active TCR, this one, but not any of the control TCRs. And equipped with this tool, now Kiwi, of course, can go in vivo and see what is happening there. And when he does a staining of either spleen or lymph node cells, he can find a population between 1% and 4% of the cells that bind these multimers. And when he showed me this data first, I was very skeptical. I was pretty sure that either it's some background or we are fishing out another interactive IL-17 receptor A, maybe IL-17 receptor C or something like that, that is expressed by a subset of gamma delta T cells. But to prove me wrong, Kiwi performed single cell sorts of this multimer stained cells, and then performed sequencing of the VDJ junctions. And when the sequencing results came back, we immediately realized then something very interesting, because all the TCRs that he fished out with the multimers were V-delta-7, exactly as the first 2 founder TCRs that I showed you in the beginning. So it became immediately clear that most likely what we are seeing here is because of the TCR specificity of the cells. CDR3 sequences are extremely diverse. We never found, I think, the same CDR3 delta sequence, although we sequenced many more cells now than just the 7. V gamma chains can be both V gamma 1 and V gamma 4, as the two founding TCRs. Mostly it's V gamma 4, again with quite diverse CDR3 sequences. And of course, to close the logical loop here, Kiwi also produced the TCRs by gene synthesis, plugged them into reporter cells, and sure enough, but at this point I was already not very surprised, he could nicely see that all of them are activated by IL-17 receptor, indicating, formally demonstrating that what we're fishing out here with multimers is explained by the TCR specificity of the cell and not something else. And of course, because V gamma 4 with delta 7 TCRs were so prominent in the repertoire, and majority of our IL-17 receptor binding cells are V gamma 4 with delta 7, we decided to check which fraction of V gamma 4 with delta 7 cells are actually recognizing IL-17 receptor. And to address that, we took advantage of a single-cell RNA-seq VDJ-seq dataset published by Thomas Winkler's lab. We identified the gamma-4 with delta 7 TCRs in this dataset, red dots here, and then we just randomly selected 15 TCRs, plugged them into reporter cells, and tested their reactivity to IL-17 receptor. And to my great surprise, all TCRs showed some reactivity. Some of them were only weakly reactive. Some of them were reactive as strong as the binder TCRs that we fished out earlier with multimers, but some degree of reactivity was always there. And this was surprising because with our multimers, we stain maybe up to 4% of total gamma delta T cells. But these red dots here, depending on the data set or depending on the animal facility, represent about 10 to 20% of total gamma delta T cells in secondary lymphoid organs. So to figure out what is the reason behind this discrepancy that we stain only 3–4%, but we expect to get 10 to 20%, TV also stained this different TCRs with different reactivities, and turns out that the weakly reactive TCRs you cannot stain, suggesting that what we're fishing out with our multimers are the relatively high affinity cells. It's probably quite a low affinity interactions to start with. We don't have any values, we don't have any SPR yet or anything like that. But it looks like with multimers, we underestimate the actual frequency of the cells, and the vast majority, if not all, the gamma 4 with delta 7 cells are reactive, and the staining and reactivity correlates quite nicely. The other thing that we did, because is quite extensive mutagenesis. This is old slide, I'm not showing you all the results here, but basically we try to replace the gamma chain, TCR gamma chain here with all possible mouse V gammas, and all of them seem to be from the first, to some extent, compatible with reactivity, so it doesn't matter so much which V gamma you have here, although ex vivo we so far found only V gamma 4 and V gamma 1 in IL-17 receptor reactive cells. And then we performed quite extensive mutagenesis in V delta 7. We found now many more than this, but we found several mutations. All of them are clustering around the same area on V delta 7 that do not change the expression levels of the TCR. It's still responsive to anti-CD3 or the anti-TCR cross-linking, but it completely loses binding and reactivity to IL-17 receptor, almost suggesting, although of course it's a speculation, that this might be somewhere here that could be part of interaction interface. But okay, this is all very exciting, but does it have any consequences in vivo? Does this interaction actually have any biology behind it? And to start addressing that, we again looked at this single-cell RNA-seq dataset, and you can appreciate that the gamma 4 with delta 7 TCRs seem to be clustering around the same area, right? It's actually one cluster here. I'm not showing you this result. And if you look what kind of cells are those seem to be Th1-like effector cells. They express Tbet, they spontaneously express interferon gamma, at least on mRNA level in the single-cell RNA-seq data set. And then we could also find a few surface markers, including CD122 and Ly6C, that function as nice surrogate markers for the Th1-like gamma delta T cells. And if we use these markers, we can nicely see that CD122 positive, Ly6C positive cells are enriched among our IL-17 receptor multimer stain cells. And moreover, if we stimulate the cells with, or if we look at the transcription factor expression, we see that these cells express Tbet but not gamma T. And upon stimulation, they sure enough produce interferon gamma and non-IL-17. Okay, that's exciting, but does it have anything to do with actual recognition of IL-17 receptor? To test this, we looked into IL-17 receptor knockout mice. This is again multimer staining of lymph node gamma delta T cells. You can see in wild type we see a nice population, but in IL-17 receptor A deficient mice we have a very strong decrease in the cells. The remaining cells in this case are still real. We sorted them, sequenced their repertoire, and it looks like they are V delta 7. This doesn't seem to be the consequence of broken cytokine signaling, because if we remove 2 out of course, more than 2 ligands for IL-17 receptor A, specifically IL-17A and F cytokines, if anything, we see a slight increase in multimer binding cells, indicating that this decrease most likely is because of the lack of IL-17 receptor as TCR ligand and not because of its function as cytokine receptor. And moreover, if we look at the phenotype of the cells in wild-type mice and cytokine-deficient mice, we see this Th1-like differentiation. They also express TBET and produce interferon gamma, I showed you before. But if we look at the IL-17 receptor-deficient mice, this effector program induction doesn't happen. They actually look more or less like naive cells. They don't express TBET or gamma T. They don't express the surface markers. They don't produce cytokines upon PMA ionomycin stimulation. But what I showed you, what I mentioned you before is that with multimers, we strongly underestimate the overall number of IL-17 receptor reactive gamma delta T cells. So does IL-17 receptor regulate only selection of these high affinity cells that we detect with multimers, or it affects all V gamma for V delta 7 cells? And to address this, for the lack of V delta 7 antibody, Kiwi had to go through a trouble of single cell sorting of V gamma 4 cells, either from wild type IL-17 receptor deficient mice, and then performing single cell PCRs for V delta 7. And then, because it's index sorting, you can even backgate and see what is the phenotype of the cells and what is their frequency. And if we first check the frequency, there is an overall decrease in all V delta 7 positive cells in IL-17 receptor deficient mice, indicating that we're not selectively losing only the multimer binding cells. We're losing the largely V gamma 4 V delta 7 cells. Remaining cells, again, are much less efficient in acquisition of this effector phenotype. And if you look at the total number of Th1-like gamma delta T cells with V gamma 4 V delta 7 chains, it's very dramatically decreased, indicating that IL-17 receptor A chain is responsible for selection something between 10 and 20% of total gamma delta T cells that we find in secondary lymphoid organs in mice. Of course, selection happens in the thymus, and so far I only showed you peripheral data, but if we look in the thymus, actually even in wild-type mice, it's a very small subset of cells that bind multimers. So there is some enrichment at the periphery, and maybe at the last few slides you will show why that may be happening. But if we look in IL-17 receptor deficient mice, what we see is actually a slight increase in the multimer binding cells. Well, slight, not slight, twofold approximate. And when we start looking at the maturation markers described by David Wist, you can nicely see that in wild-type mice, they start upregulating CD73 and downregulating CD24, consistent with what was described before, a differentiation trajectory for gamma delta T cells. But if we look at the IL-17 receptor deficient mice, these cells seem to be largely stuck in the CD24 high, CD73 negative state, and it almost feels like, although we don't have a formal evidence here, that these cells maybe there is a partial differentiation block, and maybe they just are not licensed to egress from the thymus or something like that, and only some cells leak to the periphery. To be able to understand better what's going on with the cells, because it's very difficult to study this very, very few cells, especially when you don't have the ligand and you almost completely lose them at the periphery, we teamed up with Thomas Winkler's lab in Germany, who made a TCR transgenic mouse for us. And the strategy that we developed with Thomas was the following. We took a TCR from one of our IL-17 receptor specific cells that we fished out with multimer, and then we put a full-length cDNA for TCR gamma chain, P2A peptide, and just a VDJ segment for TCR delta chain of this TCR directly into C delta locus. in such a way that we delete endogenous DNJ, so there is no endogenous TCR delta rearrangement here, and that this construct can splice to endogenous C delta. And I was worried that this will not work, but it worked beautifully. This is a polyclonal mouse here, just a wild type, and you have this fraction of the percent of total spleen size being gamma delta T cells. But if we look at our TCR transgenic mice, you can appreciate that more than half of T cells become gamma delta T cells in the spleen and lymph nodes. And of course, the cells express our VGamma 4 TCR, and even more excitedly, they induce this effector program. So TCR expression is sufficient to induce the CD122 high phenotype of the cells. And this is dependent on IL-17 receptor, because if we cross this mice to IL-17 receptor knockout background, we basically phenocopy the situation that we see in polyclonal setting. They don't upregulate CD122, and they are strongly reduced in numbers. But that numbers already is something you can work with. And now you can compare what happens in the cells that developed with and without ligand. And here I'm moving towards the end of my presentation. We don't have too much data on that. But the first simple experiment that we decided to do is what happens if we stimulate the cells with IL-17 receptor in vitro. And turns out if we do it for knockout cells, they happily respond by upregulation of CD69, by upregulation of CD25 to relatively low doses of IL-17 receptor coating. And when we do the same for wild-type cells, it looks like they tune down the threshold, right? They respond, first of all, to they need higher concentration to start responding. And they also plateau in their responses at a much lower level, indicating that somehow interaction with the ligand sets the threshold for their TCR signaling. We can actually see the same also with anti-CD3 stimulation. They're also hyper-responsive. And they become at least partially refractory to IL-17 receptor stimulation. So could it then mean that IL-17 receptor interaction is not meaningful at all for the cells later on, and what we're seeing here, this activation, is just because we override it with very strong levels of TCR signaling? To test this, and this is very preliminary data, and this is my very last data slide here, NIMI performed an experiment where she took TCR transgenic cells, either from wild-type background or from IL-17 receptor knockout background, labeled them with cell trace violet, and then transferred them to IL-17 receptor knockout mice or to wild-type mice, and saw what happens to the cells over time. And this experiment was done once. It's very preliminary. Maybe we'll prove ourselves wrong here. But so far, it looks like, first of all, if you transfer either wild-type or knockout cells to knockout, nothing is happening. They're just stuck. They don't proliferate. They just hang around there. Actually, they don't die so much. You can find them back, but they just don't divide. Now, if we take wild-type TCR transgenic cells and put into wild-type mouse, the situation changes. Already on day 5, you can start seeing some cells dividing. And by day 23, you see that a large fraction of the cells divided once, twice, maybe 3 times. So this compared to this, right, because here there is a ligand, here there is no ligand, but otherwise the cells are identical, indicates that IL-17 receptor reactive gamma delta T cells in wild-type mice probably undergo ligand-dependent self-renewal proliferation. So they're not completely refractory. They see the ligand, they respond to it, and they keep dividing. And finally, as a bonus experiment here, we can also see what happens if you have cells, TCR transgenic cells that never saw their TCR ligand. What happens if we transfer them to wild-type animals? And this would either mimic a situation when you have that gamma delta T cells sees a foreign antigen, or maybe this mimics a situation when gamma delta T cells migrate, first sees the ligand up already after egress from the thymus. And turns out they very nicely and very strongly proliferate in response to this stimulation, indicating that, you can see that as before in vitro experiments, this wild-type cells are partially refractory to the stimulation. They just undergo self-renewal, whereas knockout cells proliferate nicely when they see the IL-17 receptor for the first time. So that's where we stand. We showed that IL-17 receptor seems to be a non-clonal ligand, if you will, a super antigen-like ligand for a large class of the gamma delta T cells that utilize V delta 7 in their TCRs. And this interaction is important for selection of the cells. It actually allows to select diverse repertoire, because I told you that despite of this V delta 7, usually with VGamma 1, VGamma 4, sometimes with VGamma 1, the CDR3 sequences of the selected cells are extremely diverse. So if you would think that there could be another ligand that would be binding there, something foreign, for example, there would be a potential for the selected cells to participate in such a response. This interaction drives induction of effector program and is required for peripheral self-renewal. And of course, together, the selection plus self-renewal results in accumulation of the cells at the periphery, which is abrogated without a ligand. And together with multiple papers from Adrian Hayde and others about butyrophilin family members that function in a very reminiscent way, in a sense that they often interact with germline encoded sequences, but mostly in the gamma, but in the gamma chains, not the delta. It seems like this non-clonal interactions is a major force that shapes gamma delta T cell compartment. It can be a way to select a diverse repertoire of the cells and induce effector program in the cells. And then, of course, sometimes, as exemplified by butyrophilin family members, that results in involvement of this interaction with the peripheral responses. Here, we don't have any direct evidence for this interaction being important in the course of the immune response, but we're trying to figure out what could this mean and what could be the function of this after the cells are selected. So that's where we stand. And I think I mentioned the key people during this talk. Again, for the second story, it was largely driven by Kiwi Ye, with help of many people, including Josephine Dunst and Anya Kramer and Nimi Francis and many of our collaborators. And for the first story, it was largely work by Yuan with help from Josephine. And I think I'll stop here and take questions.